Part one.
Okay, hello everyone. Like Dr. Solis just said, I'll be giving 2 lectures this semester. The second one will be after week. So yeah, this is part 1 of the technological revolution in medicine. And I chose this title on purpose because revolution means we're changing the way we think about a certain idea or concept. And in this first lecture, I want to talk about our concepts of medicine or the role of a physician. Sorry, I'm trying to figure out this. Okay, so currently, I think how we look at medicine in our minds, in a simplified view, is that there's a disease, we use pills or some kind of medicine to fix it, which hopefully results in the disease death. And I think that in itself is something that's shifting. And similarly, we can see the role of a physician in a simplified view, which a patient has a problem, the physician treats the patient until they're no longer the patient. So this could be, the patient is cured or even just passed on to the next specialist. So the way these are shifting currently with the influx of all of this new medical technology is that, the main focus of my talk is bioengineering. And through using stem cells and regenerative medicine, I feel that this whole view of how we use medicine is shifting from, rather than finding drugs to simply kill diseases, we're trying to find cells and edit genes to actually help regenerate the body using the body's own cells and immune system. So, yeah, regenerative medicine, we've talked about this in the past in this class. I think Dr. Solis gave a talk on this early in the semester. But it's using stem cells to replace damaged tissue, and this damage could be due to age or due to injury. And I will be sharing these slides with you. So before we dive into stem cells, I wanted to talk about current applications. So I'm sure you've all heard of Dolly the sheep, probably the most famous cloned animal. And interestingly, I was surprised to learn she was born in 1996, which means she was probably born before most of us. But so Dolly the sheep had 3 mothers. One provided the DNA, another one provided the egg, and the third one was the surrogate who actually carried that embryo to term. We'll go into more detail about this, but this is just something that's currently happening. Obviously, this isn't happening with humans, but animal clones are a real thing that is currently being applied. The second one, which is applicable to humans, is stem cell transplant. So an example of this is in leukemia patients. So leukemia is cancer in the bone marrow, and the bone marrow is responsible for producing blood cells. So our current traditional chemotherapy drugs, they don't specify on which cells they attack and kill. So while they do kill cancer cells, they also kill healthy cells that are actually protecting the patients. So in order to give more aggressive treatment for cancer, while trying to minimize the damage this causes, a technique that's been used for the past few decades is stem cell transplant, in which they extract bone marrow prior to giving chemotherapy drugs, and then after the chemotherapy is taken, they then retransplant that patient's own bone marrow back into them to kind of help boost the immune system after the chemotherapy. And the last example I have that's currently being applied are stem cell biobanks. So I'll talk about this IP part in a second, but stem cell biobanks are collections of large numbers of stem cells from both healthy and diseased volunteers. And these are collected in non-invasive ways from, not super invasive ways. so from skin biopsies or blood samples. And they're available for researchers internationally, through which they can test treatments and understand disease mechanisms by having access to cells of people that they might not have access to in their home countries. So now we can look at the actual types of stem cells. So the three main types are adult stem cells, embryonic stem cells, and induced pluripotent stem cells. So we won't be talking very much about adult stem cells because they're multipotent, which means they can only differentiate into a specific limited type of cells. The other two types, which are embryonic and induced pluripotent, both of those are pluripotent stem cells, which means they can differentiate into any other type of cell in the organism. So to make that a little more clear, these up here, pluripotent stem cells, include embryonic and induced pluripotent stem cells, which means they can turn into any of these things. You don't need to know this in detail, but there are the three major cell lines are mesoderm, ectoderm, and endoderm, which simply mean the outer layer, the middle layer, and the inner layer of our bodies. So pluripotent stem cells can differentiate into any of these cell types, whereas adult stem cells, which we all have in small quantities, can only develop into specific types, like only ectoderm, only mesoderm, or only endoderm. So yeah, that's about as much detail as I'll go into for the adult stem cells. So we'll start talking about embryonic stem cells. So an example of this is Dolly the sheep. She was born through the use of embryonic stem cells. So like I mentioned before, she had three mothers. So the first one gave DNA through skin cells. The DNA she gave was in a cell nucleus, which was transplanted into an embryo of the second mother, and this embryo was implanted into the surrogate mother. So this whole process is called somatic cell nuclear transfer. And the issue with this is that while it is fairly easy to do in animals, there's limited accessibility and it's ethically complicated in humans because it does require using actual human embryos, which are questionable when it comes to acquiring them. Of course there are volunteers, but as you'll see, there's also a lot of controversy surrounding this. So in terms of using embryonic stem cells, some controversial things included whether after we mastered animal cloning, if human cloning was the next step, and the fact that obtaining human eggs requires ovarian stimulation or heavy hormone doses in women, which we don't know the long-term effects of and could be harmful. And some ethical questions include why is selling most body parts illegal but not eggs? This opens up a lot of issues surrounding like a human egg black market. And the last question, which I kind of wanted to hear your thoughts on, was should clone embryos have the same moral status as normal embryos? So, sorry, I forgot to mention this at the beginning, but I'm going to have a few slides with these kind of ethical questions periodically after I introduce some new topics. So if you guys have any thoughts on this, I think it'd be interesting to hear, but I'll just show you something else before we come back to this question. So keep thinking about whether you would classify clone embryos as the same, as having the same human and moral rights as regularly conceived embryos. But similar to this was the case of in vitro fertilization 40 years ago. So the first case of an in vitro fertilized baby was in 1978, which is only 40 years ago. And when this was being researched, the researchers were facing a lot of scrutiny, and it was being regarded as very unnatural. And this was only 40 years ago. So there's an interesting argument that I saw in a lot of newspapers that were arguing against IVF fertilization, and in which people argued that just having the desire to have a child doesn't mean you have the right to have a child, which I think today a lot of people would find not very agreeable and kind of offensive. So even though the first one was only 40 years ago, there are now over 8 million babies born from IVF, which was at the beginning received as very unnatural and unfavorable. So now I wanted to go back, and do you guys have any opinions on this? Would you think that cloned human embryos should have the same moral status as natural embryos? Because at this point, the law states that cloned embryos are allowed to be used for testing, but they must be discarded afterwards. And they also have to be, all the research in this area is privately funded because the government will not fund it at all. So it's kind of put a moratorium on this kind of research. So do you guys have any thoughts on this? Would you say, yeah?
I have a question. So can you tell the difference between a clone and the normal embryo, or are they exactly the same?
The thing is, they will develop the same. Like at the end of the day, they look the same. But of course, they only have one set of genetic material. Like they're a clone, so they don't have the mothers and the fathers. They just have one individual's genes, genetic makeup. So obviously we haven't created any human clones yet. So we don't know how they would develop as humans.
Okay.
But as embryos, they're basically the same. And I think people already put a lot of emphasis on the rights of embryos, right? So, yeah, I was just wondering if you find it questionable that we can create these human embryos and also discard them in this way, but they're still human embryos, you know? Okay, we can, we'll have some, yeah, go ahead.
I think it kind of boils down to similar concepts as if you view abortion. If you believe that everything has intrinsically a right to life, and you have to, protect that right to life and respect that right to life, then yeah, the clone embryo should have a moral status as a normal embryo. So it's the same idea as like being pro-life.
Mm-hmm.
But at the same time, if you're more towards like I guess the pro-choice side, and you believe that the embryo is just more so a collection of cells, and as long as you don't like develop it past a point, then the research, like the current way it currently is fine. So I think it leans towards similar concepts as that debate.
Yeah. So I don't see this being resolved anytime soon. So we can, of course, talk about this at the end too. We'll have time for discussion. But I'll move on to the third type of stem cells that I showed you earlier, which is induced pluripotent stem cells. So there was a study published by Takahashi and Yamanaka in 2006, which was pretty groundbreaking in terms of stem cell research. So until this point, the stem cells that we did use for research were embryonic, which, as we just saw, are difficult to navigate ethically. So in this case, for some background, they set out to create stem cells from completely mature adult differentiated cells. So from skin cells or blood cells. They wanted to revert them back to their original stem cell status. So in this study, which you don't need to know the details of, they inserted 24 candidate genes into fully differentiated adult mouse cells. they isolated the clones that most resembled embryonic stem cells. And this resemblance was based on structure, morphology, and proliferation properties. So, for example, in mice, stem cells double every 17 hours. So they isolated the clones that were most like embryonic stem cells. And after several isolation steps, they identified 4 genes, which are these if you're curious. And they found that using these 4 genes, inducing them into adult cells, they could revert these adult cells back into stem cell programming. So this was the major finding of their study, which is pretty revolutionary, I'd say, that you can take, what it boils down to is that you don't need an embryo for stem cells anymore. You can take anything from a skin biopsy to a blood sample to even cells from urine samples and turn them into stem cells. And this works with human stem cells as well. So let's look at the advantages and disadvantages of this. So, like I said, it's minimally invasive, especially when compared to taking actual embryos. It's also suitable for auto-transplantation with low risk of rejection. So this is similar to what I was talking about in leukemia patients earlier, in that if we extract the bone marrow, if we induce pluripotent stem cells from some of your cells, they already have your genetic makeup, and if you want to produce stem cells or tissue or even organs from those cells and retransplant them back into you, it has very low risk of rejection since it's already your cells that are creating this. And lastly, and I think this is one of the most exciting applications of induced pluripotent stem cells, is that they're great for disease modeling in vitro. So this means we can try treatment and therapy options that we might not have tried on the patient themselves, but we can try it on the cell culture, and we can see if those cells respond, since they have the exact same disease mechanism that you have in your own body. And it happens at no risk to the patient. So we can even try risky treatment options that we might not have directly given to that patient. So some disadvantages are that the time between extraction of these cells and transplantation is quite long. So this includes changing the genetic programming, growing the culture, etc. And this risks transferring a mutation to the patient. So there's an example of this. It was pretty famous a few years ago. So in London, at a children's hospital, they routinely did the leukemia stem cell transplant technique, where they would extract some bone marrow, give them chemo, and then retransplant their own bone marrow back to give them a boost, and it helped. It had positive reactions for years. And in 2014, they did this same technique, but it suddenly led to 4 deaths in those leukemia patients. So there was an investigation that went into this, and they found that it was at no fault of the physicians, but they found that the stem cells, they had to be frozen, kept in solution, thawed, and then reinserted into the patients, and they found that the actual freezing and thawing process is extremely delicate, which changed the actual function of those cells without changing the structure. So even though they looked at these cells afterwards before transplanting them into these patients, the function differed, which actually increased the rate of deterioration in these patients and led to their unfortunate deaths. So obviously, there are a lot of things that can go wrong that we don't quite understand yet. And this leads to the second disadvantage, that while induced pluripotent stem cells are similar to embryonic stem cells, they're not identical. There's always going to be some minor alterations, some chromosomal differences that we don't understand, because like I said earlier, there's only 4 genes that we are inducing in these cells to bring them back to their stem cell state. And obviously 4 genes might not be exactly what it takes to revert them to their embryonic stem cell status. So this is just to kind of help understand what induced pluripotent stem cells can do. It's just a recap slide that we can take somatic body cells, adult cells, induce pluripotency, which can then differentiate into any type of cell you want. And you can use that both for disease modeling and transplantation back into that patient. So, now that we're done talking about stem cells, the 3 types of stem cells, I wanted to move more into bioengineering through gene editing. So, you've probably all heard of CRISPR-Cas9. It's a modified bacterial protein, which is basically a gene editing system. Cas9 is an enzyme that induces breaks in the DNA, which can be rewritten and inserted into that gene sequence. So, you guys probably saw this coming, but we'll be talking about the gene-edited babies that were born pretty recently. And I thought it was interesting because I spoke about this last term. I spoke about it November 22nd, and in that presentation I said, quote, I don't think we will be using this technology on viable embryos for a while. And the first gene-edited babies, their births were announced 3 days after. So I think this teaches us a few things. That one, I don't think I'm a very good predictor of how fast this technology is moving. And second, that I don't think any of us really predicted that this would be happening so quickly. Like, most of us didn't see this coming. We knew it was happening, but we didn't know these babies were actually going to be born this soon, in 2018. So I wanted to give you some more details on this actual study. So the actual gene editing that went into these babies was that they altered a gene so that they wouldn't be able to contract HIV. And the reason for that is because in all of the couples in the study, there were 8 couples. Yeah, there were 8 couples. All of the men had HIV and the women did not. But all of the male participants' HIV was well controlled by current treatments. So I just wanted to add that in there because this wasn't like life or death, and it wasn't extremely debilitating for these men. And also, it wasn't guaranteed that the babies were going to get HIV because it was the men that had it, not the women. So here it starts to get a little bit tricky navigating through this study because official notice of this work was given on November 8th, and the babies were announced to be born November 25th. So obviously this work had been going on for probably years prior, but the notice was given very late. So it starts getting a little suspicious there. Something else that was suspicious was that consent forms called the project an AIDS vaccine development program. So while the researcher said that all of the couples were aware of what's happening, and I mean, I don't know, so I don't know if I can doubt him. I think it is interesting that the consent forms were called vaccine development. He also promised insurance coverage for all of the children conceived through the project until they were 18 in case of complications, which is, I mean, I'll let you decide what to think about this. And lastly, probably most surprisingly, he forged ethical review papers. And so I wanted to show you another side to this because all of these things make it seem like he was a mad scientist type. But he actually released a video defending his work, which I wanted to show you because he seems like a pretty nice guy. There's captions.
Two beautiful little Chinese girls named Lulu and Lala came crying into the world as healthy as any other babies a few weeks ago. The girls are home now with their mom Grace and dad Mark. Grace started her pregnancy by regular IVF with one difference. Right after we sent her the husband's sperm into her egg, we also sent in a little bit of protein and instruction for a gene surgery. When Lulu and Lala was just a single cell, this surgery removed the doorway through which HIV entered to infect people. A few days later, before returning Lulu and Lala to Grace's ward, we checked how the gene surgery went by whole genome sequencing. The result indicated that the surgery worked safely as intended. Grace's pregnancy was normal, which we monitored closely by ultrasound and blood tests. After birth, we again deep sequencing Lulu and Lala's whole genome. This verified the gene surgery worked safely. No gene was changed except the one to prevent HIV infection. The girls are safe, healthy as any other babies. When Mark saw a daughter for the first time, he said that he never thought he could be a father. No, he has found a reason to live, a reason to work, a purpose. You see, Mark has HIV. Discrimination in many developing countries make it worse. Employer fire people, and Mark, doctor denied medical care, and even forced his sterilized woman. Mark and Grace couldn't bear to bring a child into that world of fear. Mark's words told me something I didn't fully appreciate. A gene surgery that could save a child from a lethal genetic disease named cystic fibrosis or from an F-18 infection like HIV doesn't just give that little boy or girl an equal chance at a healthy life. We heal a whole family. As a father of two girls, I can't think of a gift more beautiful and wholesome for the society than giving another couple a chance to start a loving family. The media hate panic about Louis Brown's birth as first IVF baby, but for 40 years, regulation and morals has developed together with IVF, ensuring only therapeutic application to have more than 8 million children came into this world. Gene surgery is another IVF advancement, and is only meant to help a small number of families. For a few children, early gene surgery may be the only available way to heal an inherited disease and prevent a lifetime suffering. We hope you have mercy for them. Their parents don't want a designer baby, just a child who won't suffer from a disease which medicine cannot prevent. Gene surgery is and should remain a technology for healing. Enhancing IQ or selecting hair or eye color is not what a loving couple does. That should be banned. I understand my work will be controversial, but I believe family need this technology, and I am willing to take the criticism for them. So you can learn more about our moral value and work. We'll post up a few more simple videos.
Okay. So, I think some of you would agree. Yeah.
So I just have a quick question with the process. So if that guy just did IVF to the baby, would the baby get HIV?
He didn't, like,
Or-
he gene edited.
No, let's say he
and
did.
then-
Let's say he just did IVF. Would the baby get HIV? Because the mom doesn't have HIV, right?
There's no, like, you can't guarantee it. There's a chance the babies would have HIV.
Oh, because it edited the male's like stem or germ lines.
If it was just IVF without gene editing, then there's a chance the babies would have HIV. But he edited the genes so they won't have HIV.
Wait, why would they have HIV?
Because the dad has HIV.
Right. So I didn't know that. So HIV changes the germline DNA. Does it do that? I don't know.
It is genetic,
Right.
but it's not guaranteed to be passed on. Like,
Okay.
it said the chances were low to begin with. Like, I don't know the actual mechanism of how it's passed on,
Okay.
but it can be passed on, but the chances were low to begin
Then
with.
yeah. So now the girl has like no chance, supposedly.
Supposedly, yeah. Yeah.
So as an alternative, could they create multiple zygotes and then sequence the embryos to see which one might have had HIV and which ones didn't? And then just avoid the whole process of Cas9 to begin with?
I guess so. So it's likely that they would get some that did have HIV and some that didn't. But I don't know. That also seems a little weird to me, doesn't it? Because that would mean extracting multiple eggs and creating multiple embryos, and then discarding the ones that you didn't like. But I mean, both are questionable. So, but I wanted to show you that video because I actually watched that video before I learned about these things. So after I watched that, I was like, you know, he seems like he has a point. Like, it doesn't seem that bad. But obviously when you put it together with the fact that all of these ethical standards were violated, then it gets more tricky. And I also wanted to bring up the idea of the slippery slope that I think has been mentioned a few times. That if we start with being okay gene editing embryos that have fatal diseases, then maybe the next step would be diseases that aren't fatal, but you know, severely debilitating or limiting, like physical or mental disabilities that would really hinder quality of life. And then after that, it could just be disabilities that are really bad or that are kind of inconvenient, and it can just keep going. So just some more info on what happened to him. He hasn't been seen since his publicly seen since his announcement. There were different stories on this. He's either on house arrest or hiding in the university dormitory. But either way, no public appearances. So some questions I wanted to pose to you guys were, should these babies know that they were the first gene-edited babies? Because they're going to be obviously under the public eye and being medically tested throughout their lives. Obviously Lulu and Nana are fake names, they're not their real names. And do you think the researchers should be punished? And how about the parents who went into this knowing that their embryos are going to be edited? Like, how would you deal with this? How would you figure out how to make the legislation to deal with this kind of thing happening? Does anyone have thoughts on that? 10% participation. I can give you time. Yeah, go ahead.
Just a very quick first thought. I would say that he probably doesn't deserve the amount of flack he's getting, because most of his current ethics are probably the result of, I suppose to say, the usual work ethic in China, where he's doing most of his research. It's pretty easy to just avoid most of the normal ethical responses, and there's, from what I've heard from friends and family, there is zero ethical standard for most things there. So.
The government also publicly condemned him. And
but that was after.
Well, yeah,
it was
that's true.
released, and everybody else got mad. So they're like, "Oh yeah, we gotta get mad too. Otherwise, people are gonna get mad at us."
So it's a conspiracy.
Could be. Who knows?
Wait, someone here? Yeah.
Oh yeah, I think just on the question of how to legislate around it, at some point there will probably need to be some kind of international standard to meet. Like we see with traditional human rights, there's the Geneva Convention of Human Rights. And this field is obviously not kind of at that level where everyone has access and everyone accepts the principles around it. But it's problematic when you have a private institution in one country kind of setting the norm of what that should look like. Also problematic, as you mentioned, that it was a privately funded project and the government didn't even know about it to begin with. So to legislate, people first have to know about it, and you have to agree on what those standards are before you go and do it.
Yeah. So that would entail that someone who researched like this without gaining approval for it first, they would face some kind of consequence.
I don't know what that consequence would be, but it's problematic to just kind of give free rein to let anyone who has the
Yeah.
resources go experiment.
I think that's true, especially considering that CRISPR-Cas9 is so ubiquitous in labs around the world. It's pretty easy to get your hands on and pretty easy to actually manipulate that science. Did anyone else have any final thoughts on this? Yeah?
I have a question. How did IVF become like okay? Like I'm guessing was it like one or two scientists or institutions that like performed it and then everyone was like, oh that's
There were two scientists who in 1978 they research had been going on it for a while, just like research on human embryos has been going on for a while, but these two researchers, they have a pretty parallel story. They also couldn't get public funding and government funding, so they also acquired private funding to, create the first IVF baby. And it was very controversial and seen as unnatural, but the ethics just evolved. Like people had that conversation, lots of people were angry about it, lots of people were like, maybe it's okay if these people can't naturally have children, why does that mean they just have to be childless their whole lives?
Mm-hmm.
And it just evolved over the years to how it is now, where it's no one really
Yeah, it's
cares that
like-
much anymore.
maybe this is the starting point.
Yeah.
Sometimes you need like a drastic rebel or something to
Yeah, that's why I thought it was interesting that in his video he specifically said he knows this is going to be controversial and he's willing to take the criticism for it. So yeah, that's where it gets confusing on where to draw the line between this is inspirational science versus is this safe? Yeah.
What if like the parents approached him first? Like, would it be okay if it was their idea, not his? Or like, if like he just did it to himself, would it be
Like his
better?
own children.
Yeah, or like to like himself, like he like edits his own genome or something.
Well, for the first part, I think it's still the responsibility of the researcher. Like, if, like, Dr. Solis, you're a physician. If someone came up to you and asked for something that was not very ethical, and if you did it, you would still get the blame for that, right?
Yeah, I think it isn't quite as simple as it may seem that you can't deny that progress was somehow made here, right? And people started thinking about things that they weren't thinking about before and so on. So it's never completely black and white. But I think part of the reason that we talk about this is that as time goes on, these situations are going to become more and more complicated and like with no easy answers. But somehow, the human species, our society is going to evolve and get better on average through these things, but there'll be some things that are definitely negative. And this guy had U.S. collaborators or U.S. scientists who knew about what he was doing, and they have also been impacted. Like their institutions are going after them. What did you know, and when did you know it, and why did you tell us, and stuff like that. So it is bound to be a kind of combination of science and politics, and some of it ends up in a realm where there are just no easy conclusions. You can't have bullet points about do this, don't do that, and so on. But eventually it'll come exactly to that there will be guidelines for this, and international bodies will agree on it, and so that's what will eventually happen.
Did anyone else have anything to add? Okay, so now we'll be shifting our focus a little bit. So up till now, I've been focusing on a lot of bioengineering that's applicable to humans. But the next thing we're talking about is applicable kind of exclusively to animals, and specifically lower animals and insects that reproduce pretty quickly. So gene drives are a type of bioengineering technology that can propagate a specific set of genes throughout an entire population. So I'll explain this a bit further. So as a review of how normal inheritance works, even if we gene edit one, like the male in a pair, and the female is left normal or wild type, if we follow normal inheritance patterns, then only 50% of the offspring will get it. So it won't propagate very widely, it'll remain pretty contained. So how gene drives work is CRISPR-Cas9, which we've just talked about, is used to edit a gene. So in this case, it's this blue part, the cargo. We stick this specific thing that we want in this gene. We insert it into the drive. But the difference is that we also insert the actual CRISPR-Cas9 system into the genome as well. So that's this pink part. So in the homologous chromosome, which is the normal or wild type one, the CRISPR-Cas9 actually induces a break in that one, which we haven't touched. And because of the break, the damaged chromosome ends up copying whatever is on its homologous chromosome, which ends up creating two drive chromosomes, which means it went from heterozygous to homozygous. So rather than having a wild type and a drive, it now has two drives, which means all of its children will also have that drive gene. So this is how the gene drives would theoretically work. We just edit one parent again, but because the actual genes edit themselves within the genome, it is passed down to every single child, which means eventually, as these edited organisms mate with normal wild-type organisms, every single member of that population now has the edited genome. So this, we haven't applied to any species yet, it's still theoretical. But basically what this would suggest, I found this really interesting because there's lots of talk, when we talk about gene editing, people like to bring up the whole playing God thing. But in this case, I feel like it's more, this is actually more applicable because we can use this technology to manipulate entire populations. And another thing I found interesting is that we also have strategies to reduce the likelihood of resistance to this gene. So, yeah, like I said, these are currently happening in lab. Like, this is happening, but we haven't actually applied it to full populations. And we have two types of gene drives right now. Standard drives, which means we just insert a specific gene that we want into this organism. So the example that has been already tested in lab is eliminating malaria from malaria-carrying mosquitoes. So if you eliminate the gene that carries malaria in just one or just a few mosquitoes, and then let them breed naturally with regular mosquitoes, all of their offspring eventually will have that gene. And in the mathematical scheme, I believe it said, yeah, the model suggested that if this was released, that within 1 year, the full global mosquito population would be gene edited to not be able to carry malaria. So the second type of drive is suppression drives. So in this, they would actually manipulate the genome so that after breeding, the embryo could only be male. It could not be female. So because all of the children would be male, that species would die out within a generation or two. And the theoretical application for this would be to remove invasive species and allow our native species back into that ecosystem. But this is difficult because if these gene-edited organisms somehow found their way back to their native population, it would also wipe out that whole population. So this isn't being used yet. I'll skip over this because I don't think we have the time to go into detail. But so in terms of how to manage these things, I think it's interesting because things like fruit flies or mosquitoes, even when they're in lab, they're quite difficult to contain. So if you if you added a gene drive into some of these organisms and they actually were able to escape your lab and enter the wild population, it could lead to huge devastating effects. So this is another one of those things where our legislation and our rules on how to work with things in the lab tends to lag behind and then catch up after something has happened, similar to what we were talking about with Lulu and Nana. So this is another one of those things where we need early engagement, because I don't think most people gene drives haven't been sensationalized by the media yet. They haven't been picked up as a buzzword like designer babies has. But it's one of those things that is coming up and does have pretty widespread implications. But again, this is not in humans. So the last thing I wanted to talk about was gene editing and cloning together. So you may have heard of this. This only happened last month, but this is also in China. But there were these macaque monkeys that were gene edited and then cloned. So they gene edited a group of monkeys to knock out this specific gene, which is responsible for the circadian rhythm. And so they knocked out that one gene, and then they chose the one monkey that exhibited the most severe disease phenotypes, and then they cloned that. So by disease phenotypes, it refers to reduced sleep time, elevated nighttime locomotive activities, dampened circadian cycling, increased anxiety and depression, and schizophrenia-like behavior. So there's actually a video of these monkeys available online. I won't show you right now because it is I think it's a little difficult to watch. When you don't have the background, it just seems like baby monkeys who are just, walking around and crying and stuff. But then when you realize that they're probably, they have, these schizophrenia-like symptoms, then it turns a little questionable to think about, because they chose the monkey that displayed the most severe disease, and then chose to clone that into these 5 baby monkeys. So this again comes with its advantages and disadvantages. So the researchers themselves are saying that if we can custom make these organisms that are closer to humans, we could reduce the numbers of monkeys used in labs. Plus, we have that specific disease that we want to look at, and we can figure out how it works, how to treat it, because all of these monkeys have the exact same genome. So we can apply different treatments to them and see what works, without having to deal with other factors that may arise by having different genomes. At the same time, critics of this were saying that these monkeys are just seen as tools rather than actual living animals. And in this case, they weren't actually even created for a schizophrenia study. They were just created to see what would happen. So, Gary Goldstein, in his last week in his class, he mentioned anticipatory ethical systems. So I wanted to bring this up. We've been talking about this throughout the course, I just haven't said anticipatory ethical systems, but I think it's very important to establish these guidelines and the boundaries of what we as a society are willing to explore and which lines we're willing to cross before we actually cross them. So I think that like we were talking about with the gene-edited babies, these revolutions, they are really integral to the advancement of science. It's 100% true. But I think it's also important to look at our values as a society, and if we're actually willing and at the point that we can accept that we're actually editing embryos before they're born. So yeah, these are just some of the other questions that we looked at, like what are the consequences for breaking the law? Would we punish researchers? And what about parents? And what about the babies? Obviously we wouldn't punish the babies, but I mean, what would happen to them as they grow up? Are they allowed to know that they were part of this experiment? And how do we shield them from the medical testing and the public fame for weird reasons that will inevitably follow them throughout their lives? Which is what happened to the first IVF baby. She did remain anonymous for a while, but for much of the later part of her life, it was known that she was the first IVF baby, so it was pretty heavily publicized, her life. And like I mentioned before, there's the question of the slippery slope of genetic modification. As if parents realize that their child is going to have a fatal disease, they might elect to have it changed. But you know, these things, maybe if you have a little bit more money, you can also afford to edit your embryo who has like something slightly wrong with them, but you know, wouldn't fall in the fatal category. Yeah, so just to recap what I talked about today, because it was, I think, a lot of different topics. We talked about 3 types of stem cells and gave some examples. And gene editing, which included CRISPR-Cas9 and gene drives. And finally, the gene-edited encoded monkeys, which use both embryonic stem cells for cloning and CRISPR-Cas9 gene editing. So to conclude, I just wanted to go back to this conceptual shift in what medicine and physician means. So in this case, this is our current model of what medicine is supposed to do. And I think that will change rapidly in the coming decades to a model that's more based on using our own cells and our own immune systems and using regenerative medicine to focus on more enhancing the body rather than killing the disease. And so here are some examples of how that could work. The first one's actually already in use. In terms of in macular degeneration, it involves the degeneration of retinal cells. And we're already using stem cells to produce new retinal cells and transplanting them into these patients. These two aren't in use yet, but they definitely have the potential to be in use because a lot of these ubiquitous diseases like diabetes and Parkinson's, hypertension, kidney failure, they have specific cells or specific tissues that can be regenerated using stem cells. and transplanted into the patient, which would be pretty revolutionary since these diseases are very wide. They're present in a large part of the population. So similarly, in the concept of physician, rather than having a physician that has an issue, treating them until they're no longer a patient, you might switch to a patient that doesn't really have issues right now, but we can enhance that patient to make them better. And this doesn't necessarily mean cyborgs or things like that, but for example, things like preemptive stem cell treatment. So right now, osteoarthritis is something that older women get pretty commonly. I think it was 85% of women 70 plus have osteoarthritis, which is a pretty big percentage. So in things like that, or things like heart disease, if we could predict this coming based on family history or clinical symptoms, and we could actually help prevent it before it actually happened. So essentially enhancing before we have the issue. This could create a huge shift into what the role of a physician actually is. Instead of going to them when you have a problem, you could actually reduce future problems by going to them. So yeah, that's the end of my talk. And if you guys have any thoughts or any questions, I would be happy to take them. And also, I've included my email. I'll send you this presentation in case you wanted to email me about your ideas about papers or presentations, because I wanted to remind you that the topic deadline is March 12th. So yes, feel free to do that. So yeah, any questions or concerns?
I think I just missed the detail where you're talking about between standard drives and suppression drives.
Yeah.
So how are standard drives selected for? Because you said in the end the entire population has it. So like why is it like favorable for the organisms to like keep that drive?
That's where it gets interesting, because it doesn't have to be favorable. Like it could be in suppression drives, it's literally killing off the population, right? But that's the thing, it's not regular evolution, it's not survival of the fittest. Because we have that CRISPR-Cas9 inside the genome, it'll edit and add in that gene no matter if it's good or bad.
I see.
Like it could be disabling that organism, but it'll still keep propagating.
That makes sense.
Yeah. Any other questions?
So this puts a lot of different things together, I think, in a very useful way. You can imagine what things will happen in terms of the politics of this. There'll be something, you kind of alluded to this, kind of circus freak or circus sideshow created of a new human being that everybody knows about with some unusual feature, and that person's life is going to be very strange because and so does it. Is that logical to ever do that, to create a human being for some specific purpose, just kind of as a demonstration, and then that person is alive? And then the alternative question is, how do we make progress in this area without ever doing that, without embarrassing a single human, without violating any of what seems like the obvious ethical norms that we should all be following? You know, how are we gonna get from A to B without some people breaking the rules? Yeah, it's a real puzzle.
Yeah, I think it's like I myself, I think my opinion is a little hypocritical, because I think that it was a little too soon to gene edit real babies, and I think that it would have been better to do it when we had actually had that discussion as a society and had more rules in place, but I don't think it would happen naturally.
Yeah.
I think this is kind of needed to stimulate that conversation.
Yeah.
Just a minor add-on to that. From history, it looks like people who actually make innovations don't wait for society to catch up. Say people like Galileo, or rather, who was the person? Crap, I don't remember anything from Physics 114, but the guy who thought the Earth was in a geosynchronous orbit instead of heliocentric. My point is, society will catch up in any case. So.
Yeah, I agree with that. I don't know, I just think it's difficult. It seems a little, even if the parents knew what they were going into, it seems a little unfair to the parents and to the children, since they are going to be the center of this controversy, like, for their lives, their whole lives. But, I mean, if they were willing to do that, and they knew going into that this would happen, I mean, it's inevitable, then I guess it's their choice. I can't say anything about that.
I think ethically, of course, it's like gonna get judged, but personally speaking, I respect what he did. Like, I think we always need a catalyst, or else like there's no need, right? It's not in the front of the mind for the scientists to even care to talk about it. So like you said, it's like a catalyst to talk about it. And I was thinking like maybe in the future, I don't know when in the future, but like almost like vaccines, although they're very different things, like the majority of the population, there's like a list of diseases where you can like check off what genes you want to like get rid of.
Yeah.
Yeah, just kind of on that as well. I think it's probably good that the first known use of it was generally for good. Like, designer babies are a big topic, so at least it wasn't just like a superficial kind of impact that it had. So I think it's good that the first thing was at least with like generally accepted moral intention, that it was like used for good instead of for bad.
Yeah, I think it's interesting to compare it to vaccines, though. So, just like we know that kids now who are vaccinated aren't going to get these, life-threatening diseases, we can also be sure, since from when they're an embryo, we can be sure that they're not going to get these genetic diseases either. But then we have to figure out which diseases that applies to, right? And how do we tell the severity of the disease?
Mm-hmm. It's like Dr. Solis said, when do we do it for health, and then when do we start determining what they should be in the future? Like, obviously, like a circus clown would have an advantage if they had like longer arms or whatever, but then we're determining what they should be rather than like just health-wise in general should be more.
Yeah, it sort of trivializes our humanity in some way, but there's the potential to make humans much better, amazingly better. So, I don't know. It's a very interesting, perplexing thing.
So you said like all this like gene editing and stuff could be like a slippery slope where like people would like want to like fix like small things and stuff.
Yeah.
But then like wouldn't also like the conceptual shift of like medicine also be like a slippery slope where like, you know, patients just want to be like enhanced, like it's not like necessary, but like it's just like, oh, I want better eyes and stuff. So like is that like bad then, or what do you think?
I think that's, for sure, I think that's already happening. I don't remember whose lecture it was, but where they had those implants for like Wi-Fi, I think, and all of these random things, or they have those chips at workplaces that like you use as a scan card. But I mean, I think that's just unavoidable. Like, just, I think similar to how there's abuse of traditional medicine right now, of traditional drugs right now, there will continue to be abuse of whatever our future medicine is. But it's, again, I don't think it's one of those things that will take over society. Like, I think there will always be people on the fringes who take things really far. But I hope that most of society would remain in that middle ground. That, I mean, obviously the middle ground is also constantly moving, but yeah, I think everything is a slippery slope when it comes to that. Yeah.
And one more thing. Even with all the current ethical standards and discussion about the semantics and everything else related with ethics, it still didn't stop that guy at all.
Yeah.
So,
Yeah. There's always going to be people that just do it, even if it's established that you shouldn't do it.
yeah. I guess there isn't much that can stop those people anyways.
Yeah.
And they're the people driving things forward, so will ethics always be trailing in the shadow of these kinds of people?
I think legislation for sure will always be lagging. Like, in most cases, it's always behind. It's always reactive to something happening. Obviously, it'd be nice if that weren't the case, but up till now, that has been the case. But that's also why I wanted to show you guys that video, because I just wanted to highlight that these aren't like insane people who are doing this. Like, he's obviously a really brilliant scientist, right, who seems to care a lot about the people in his study and the children that were born from it. So it's, when we make these ethical judgments, we also have to take into consideration that the other side also thinks they're being completely reasonable and rational, right?
So, you know, there is an alternative you can think of where machines are in charge of the world and much more intelligent than we are. And there, the laws could be changing more quickly than we can ever understand, right? So that would be very troubling maybe for us, but it may turn out fine for the world that as soon as these super sentient machines realize that something in the laws governing the world need to be changed right away, they change it, but they realize humans are too darn stupid to understand, so why even explain, you know? So at least in the world of today, we can feel frustrated that laws change so slowly, but when they do change, we can kind of figure out why they went the way they did. And there will be a time when we're kind of nostalgic for that, when we could still understand. And if you think that has no human relevance, so I trained at Johns Hopkins, and then I came here as chair, and the departmental administrator looked me in the eye and said, I have no idea how you guys can go and lead departments elsewhere because you have no idea how things work here. And I realized she was absolutely right. There were so many layers. It was such a complex world there. For instance, the big leadership positions, their presentations of the candidates would be secret, that only the committee deciding who these future leaders would be knew even that these people were coming. I mean, it's like something out of a movie, right? You can't believe real life would be like that, but that's the way Johns Hopkins was. And I did exist there for many years, 72 to 87 before I came here. And I learned that the leaders I knew never really understood why the institution did the things it did, that there were so many layers between them and the top leadership, So it really already exists now, but there'll be a time when there's a lot more of that, when things are moving in a favorable way, but you are so far from understanding why this particular thing happened and who decided that, and so on. So, and one of the reasons that I came to Canada is it seemed to me that the systems, at least in Edmonton here, there were many fewer layers, and you may not like everything that happens, but at least you can figure out sort of why it happened. Yeah, so anyway. Okay, we got 7 more minutes. Any more questions?
I think it's kind of a paradox that like society wants specialization, because
Yeah.
specialization is always the best option, I think.
Right?
But then if it's so specialized, not everyone can understand, and like you said, so many layers, but everyone's specialized because they know what's best, right? Because they understand.
Yeah. But if you think of then overarching concepts, like people are talking about, explainable AI, right? We need to know why the AI comes up with the algorithms that it does. Well, that's a challenge for machines, just like any other. Find a way to explain what you're doing to humans so they can understand it, right? So there will be something like that. It probably won't be perfect, but even when machine intelligence is much greater than ours, there'll be some sort of simplified explaining. These super smart overlords will get us some kind of message that kind of indicates why things are, the way they are. or at least in a favorable outcome, that would be the case. There's also the outcome where the people in charge, ignore us completely and there's no conversation whatsoever. Sort of like us and ants or us and grasshoppers or something like that. So, any other questions? Yes?
Just a comment. If you look at something like epigenetics, obviously it's the study of heritable changes in phenotype without specifically changing the DNA sequence. And although it doesn't necessarily operate by the same mechanism of actions such as gene editing technologies like CRISPR, you can theoretically get the same results. So like, for example, if you look at something like gene silencing, through mechanisms like methylation and deacetylation, you can silence certain genes. You could theoretically do the same thing with CRISPR, I believe, but I just think it's funny how if you look at it from a perspective of epigenetics, it's considered more ethically acceptable compared to if you're directly editing the genes. So I think it's just funny, depending on whichever perspective you take or whichever mechanism you approach it from, is met with different perspectives and different forms of backlash.
Is it seen as more acceptable? Like, I haven't read many studies.
Well, I mean, it is a relatively new field, but for example, in cancer treatments, epigenetics is new and emerging. And it's being studied extensively even here at the U of A, like at the Lee Fashing Center. But in that case, it's seen as this revolutionary technology, and everyone seems to want to be on board with this new revolution. So, yeah.
I'm not too clear on epigenetics, but I think maybe, I'm just guessing it'd be more ethical because it's not like permanent. Like whereas like if I edit a gene, it's like within the genome now. Whereas epigenetics, I could demethylate or I can methylate it again, so it's like kind of reversible. Technically, like gene editing is
Yeah.
also reversible, but it's like, I guess it's much more difficult. I'm guessing. I've never like, I would think.
Like,
'Cause
the
it's-
thing is, it's heritable too.
Oh, it is heritable
Yeah.
too. Okay.
I thought like epigenetics was just like the state that you're in like at that time, so it's like more like natural, I guess, than just like editing it straight up with like machinery.
Like you're talking about like the switch and flip, right?
Yeah.
Turning genes on and off. But in response to that, I think that gene editing using CRISPR to, you know, take out things and add things, I think everyone was on board with that too, until it hit this like babies, like real babies point. But I think up until then people were pretty excited about it. So I think it's more about the actual application of it rather than the technology itself. Any other comments? Okay, well, thank you for coming and for listening.